Yes, so it is a pleasure to host today in our Bar-Ilan University Vision Science Seminar, Dr. Alexander Göttker from Justus Liebig University in Gießen. Alexander did his BSc in psychology in Justus Liebig University. I hope I'm pronouncing this correctly. In his master's, he investigated the interaction between saccadic and pursuit eye movements and their effect on motion perception under the supervision of Professor Karl Gegenfurtner. And then he continued in Karl's lab to do his PhD, where he studied the interaction of saccadic and pursuit eye movements for oculomotor control and perception. He later did an internship with Meta, Facebook, in the US for 6 months. Today, in his postdoc, he continues to investigate these exciting topics about the relationship between eye movements and perception, and his research is also funded by a grant from the Facebook Reality Labs. He already has several publications in top leading journals as Current Biology and PNAS, on top of additional publications in vision-related journals. He also has 1 patent on his name with colleagues. He was awarded the Best Dissertation Award from the German Psychological Society. So without further ado, I'd like to welcome you, Alexander, to our seminar to hear about your exciting research, and thank you for joining us.
Okay, yeah, thank you very much for the invitation. I will quickly try to share my screen. Okay, so it works. Okay, yes, yeah, thank you very much. It's a great pleasure to have the chance to talk about my work in this nice talk series. And I will be talking today on eye movement research that is spanning typical artificial experiments where you typically would just look at dots, to eye movement behavior in more natural scenes, for example, dynamic videos. And the overarching question will be, how much can we learn, or how well do the results that we obtain in our typical artificial experiments actually generalize to understand how natural behavior in the real world is happening? But before we get to that question, let's take one step back and think about first for a second, why are eye movements actually interesting, or why do we need eye movements? And if you see a picture of such a nice castle here, you have the impression that you can see the whole image with all of its detail basically in one glance. So all across the image, you have one continuous percept. However, because of the setup or the wiring of our visual system, this is actually not the case. Because while you have the highest possible amount of visual information in the fovea, so the central part of your visual fields where you're currently looking at, resolution is actually drastically decaying towards the periphery. So instead of having the whole image with all of its available detail at one glance, what you actually almost always is doing is you move your eyes around across this image and then scan important parts of the image and piece together your visual percept. And that already shows you why eye movements are so interesting, because what we're currently looking at is the most interesting feature, or the most interesting feature that our brain thinks is really the most relevant one. So therefore, a common metaphor that is used in eye movement research is that the eyes can provide some sort of a window into, for example, perception, cognition, different brain mechanisms, the mind, and many more things. However, if you look at most of the literature on basic oculomotor control, where we always are aiming to understand natural behavior, most of these experiments look something like this. So you're usually sitting in front of a computer screen where you would, for example, have a gray screen, a gray background, and then there would be, for example, one target appearing. So this is a very famous called pro-saccade task. So your only task is you're fixating in the center, then this peripheral stimulus appears and you will make an eye movement towards it. And these paradigms were so successful because they can already tell us a lot. So here, for example, you could vary the contrast of this target and see how that affects, for example, your sensory processing reflected in how you move your eyes towards these targets. Another famous paradigm is the so-called anti-saccade task, where, for example, the setup looks almost identical. However, now, instead of looking at the dot, you have to actively suppress your eye movement and look into the opposite direction. And the error rates in these kind of tasks have been highly successful and linked into different cognitive mechanisms, psychological impairments, and so on. Third important paradigm I want to quickly mention is the so-called saccadic adaptation paradigm, where again, you have a single target on the screen, but as soon as you make your eye movement, the target is shifted to a different position. And since visual sensitivity usually is reduced during your eye movement, you might not even notice the shift, but over time, your eye movement amplitude will adapt to that. And these have provided some interesting insights into learning mechanisms in the brain. And the last paradigm I want to mention is that while all of these others use usually static stimuli, so just a static dot, for example, on the screen, there have been also studies looking at moving dots across the screen. And these were usually made to look into how a continuous sensory input can be transformed in a continuous motor output. And the one thing that all of these have in common, all of these paradigms, is that they look quite artificial. And this is not something that is only happening in eye movement research, but across vision science, we often basically try to boil down our stimuli and create artificial stimuli to investigate different questions. And arguably, these look vastly different than how our natural world looks. So why do people use them? Because they have some very obvious advantages that are really important. So one of the advantages is that we have full control about all the relevant parameters. So everything we have on the screen, we are in control of. We can isolate certain factors, for example, contrast of the target or color. And we can use these to isolate specific mechanisms and really try to understand how these work to get like this window into the mind. However, that begs the question, so how well can we actually generalize from these results if we then want to understand more natural behavior? Because while in theory that could work, there are some obvious differences as well between how these typical artificial experiments are set up and how we would behave in the real world. So, for example, while in these artificial experiments, usually you have some sort of a trial structure. So you will make one eye movement and then, for example, the next trial starts. In natural behavior, you have a much more complex and continuous response. So you will mix different types of eye movements to track a moving target. There are much more things going on, for example, in terms of eye and hand coordination. So in the real world, behavior is usually much more complex. An additional thing is that in these artificial setups, you don't really have a lot of expectations what's going on. So you don't really understand how these dots are working. And if you wouldn't introduce any statistical structure, for example, in your experiment, there's not much really experience that you would have with how these stimuli should behave. Whereas in the real world, again, we have a lifelong previous experience, for example, on how lighting typically behaves, how we would interact with objects, how objects would behave if they hit each other, and so on. And a third difference that I want to focus on is that while the artificial experiments have the big advantage that we can isolate certain factors, natural stimuli are much more complex. So they do not only vary in contrast, but also in color at the same time. There's a much more complex statistical structure of the world than we typically have in artificial experiments. And along these three different topics, I try to structure my talk by introducing examples of our work where we try to tackle each of these points to see how well or what we can learn additionally if we, for example, allow for more complex behavior. And this first study I will be telling you about is focused on how you coordinate different types of eye movements, and in this case, saccadic and pursuit eye movements. And this is work that I have done together with my PhD supervisor, Karl Gegenfurtner. So the two main eye movements that we are in voluntary control over is saccadic and pursuit eye movements. So for those of you who are not familiar with them, saccades are these quick jumps of the gaze that you can see on the left. And these are the eye movements that you, for example, would make if you look around an image, like the castle that I have shown to you in the beginning. And saccades are typically studied, for example, with stationary dots, or while looking, free viewing images, or while reading, for example. And there are these very fast discrete jumps that allow you to basically bring any point of interest quickly onto your fovea. Pursuit, on the other hand, are these continuous rotations of the eye that allow you to bring and keep an object that is moving onto the fovea. And therefore, they're also mostly studied with moving things. So for example, moving dots or in dynamic videos. If you look a bit into the literature, for example, here we already talked about VSS. This is the program from last year, so it's one of the biggest vision conferences. Saccadic and pursuit eye movements usually would be discussed in different sessions. If you look into textbooks, they usually would be treated in different chapters. And there is often the assumption that they might be different subsystems in oculomotor behavior. However, in the natural world, they always interact. So here you can see a movie now with a lot of observers watching the scene. And then at some point you can see that all of the observers converge on one object, which is actually a flying duck that is flying across this river here. And you can notice two things. First, motion is a very salient cue. So basically, as soon as the moving thing appears, almost everyone is looking at it. And second, that all of these people usually will make a saccade towards the moving object, then will pursue it. And then also during the pursuit, there will be additional corrective saccades. And contrary to that view of the classical separate systems for saccadic and pursuit eye movements, already Rich Krauslis and the group from Philippe Lefebvre have proposed there's actually a lot of shared information and also comparable neural underpinnings of these two types of eye movements. And if you're more interested in that, Carl and I wrote a recent review article where we reviewed the recent literature about this topic. So let's focus a bit on how saccadic and pursuit eye movements usually work together. And for that, I will introduce you to a classical paradigm, the so-called step ramp paradigm that was invented by Rashbas in the early 60s. So here, again, you're fixating in the center on this black dot, and then a peripheral target would appear. If that would stay stationary, what usually would happen is that you would make a saccadic eye movement to this target location. However, if as soon as the target steps into one direction, it actually starts to move towards your initial fixation, you would not react with a saccadic eye movement, but would typically already make a pursuit eye movement in the direction of the target movement. Thus, if you are able to predict where things will be going, that, for example, could lead to a suppression of saccadic eye movements. And to show you that in a bit more detail, I have some actual data here. So this would be a single trial where on the x-axis you see time, and on the y-axis the position of the target and the eye. So the target was the dashed line, and you can see that at 0, it basically steps into one direction, which in this case would be the left, and then it moves across the screen, which is basically reflected in the slope of the line. And the eye response basically also starts about 150 to 200 milliseconds. You can see that the eye also starts to move. It matches the velocity of the target, and you can just have a pure pursuit response to that target. However, if you make the step slightly smaller, what will happen is that still you would have initially a pursuit response in the direction of the target movement, but since you're lagging behind the target, you actually will make a saccade to catch up with it and then keep tracking it. On the other side of the spectrum is basically if the step would be too large, your eye still would start, but then you would perform a saccade backwards. So you basically jump with your eyes back to wait for the target and then keep tracking it. And an interesting side note here is that although these targets all move at the same velocity, we have studied this in more detail, and we found that actually how you perceive the speed of this target changes depending on how you move your eyes. So trials with additional corrective saccade are perceived differently than trials with pure pursuit. But for now, we will focus on how these two work together. And you can quantify the coordination in something that is called a smooth zone. So on the x-axis here, you see the target crossing time, which in this plot would be the time after the target after the step, it crosses over the initial fixation location. So in the center here, for example, you can see that with these slight grayish vertical lines on the x-axis, there is a target crossing time of 200 milliseconds. And you can make it longer or shorter. And what you see here is now different target crossing times plotted against how likely it is that in these trials there will be an additional corrective saccade in the beginning of the trial. So what you can notice is that, first of all, the oculomotor system is probabilistic and not deterministic, because there are some trials where in 50% of the cases, you will have an additional corrective saccade, and in the other 50% of the cases, you will just have a pure pursuit response. And there seems to be a very dominant shape, which is that there seems to be one stimulus or a center of the smooth zone, so to speak, where there's the minimum number of saccades, which for this participant, for example, is roughly at 200 milliseconds. So here, they are most likely to not have any additional corrective saccades during the tracking. And what we did now is we actually measured this coordination of saccadic and pursuit eye movements for a large group of observers. So we did that for over 50 people, and we not only did it during fixation, but also during pursuit. And what you can see is that, first of all, the correlation between these tasks is very high, suggesting that there is stable individual differences in where basically the minimum of this zone is. So what is the optimal pursuit stimulus for this person? And there's actually a large difference. So for some people, the center is close to 150 milliseconds, whereas for others, it's more than a twofold increase. So there, the center is at like 350 milliseconds. That means that there is a large difference in how different people coordinate these two types of eye movements. And that basically would mean that if I would show one type of trial to one observer, I could basically always see pure pursuit, whereas if I would show exactly the same stimulus to another observer, they would maybe always have additional corrective saccades. So we were wondering, where are these differences now actually coming from? Or can we somehow find an explanation for them? And for that, all of the observers that we measured here actually also completed a battery of different oculomotor tests where we measured saccadic and pursuit performance in isolation. So basically like in these classical experiments. So here you see one example of this. So the center of this zone, the minimum here, basically correlated, for example, with pursuit latency measured in a separate task. And we did that across a bunch of measures, which I want to show to you. And we can focus for pursuit, which are the left graphs for the beginning. And you can see here that the center correlates with pursuit latency, as well as with pursuit gain, which is a measurement of accuracy of how well you can pursue a target. And the logic of this correlation basically is the better you are in pursuing, reflected in a lower latency or in a higher gain, the more likely you are to use pursuit to catch up with targets that move away from you. And interestingly, the center here was not only correlated with pursuit measurements, but it was also correlated with saccade measurements. And the measurement that was interesting was that it was correlated with the accuracy of saccades to moving targets, but not with the accuracy of saccades to stationary targets. So the correlation with saccades to moving targets again basically reflects the same direction. So if you are good in making saccades to moving targets, then you're again more likely to use them to catch up with moving targets. And we also had some other conditions where we, for example, measured their perceptual ability to discriminate different velocities or different position steps, and there we didn't find any correlation. So what we can say from this is basically that how you coordinate saccadic and pursuit eye movements is tailored to your own strengths. So people with better pursuit, they use it more to catch up with moving targets, whereas other people who might have better saccadic eye movements to moving targets, they rely more on them. So if we look at that in a summary, we can see that on the sensory level, there have been shared information between saccadic and pursuit eye movements. But if we focus on the motor aspect, how you coordinate your different types of eye movements, largely affects on your own strengths. So you kind of have an implicit knowledge about that you probably have gathered across your lifetime on how you can move your eyes. And based on that, you somehow tailor your coordination of these different two types of eye movements. So that brings us back to our initial question. So what can looking at more complex continuous behavior tell us? So in these typical abstract experiments where we have these trials where we only measure one eye movement, we never really can look at the coordination between them. And if we go into more complex continuous behavior, we can see that, for example, how you behave there is actually can be different from person to person, and that these differences are usually also related to your own strengths. Okay. We'll take a quick break. Okay, so that brings me now to the second section. So here we will now focus on how your previous experience with the world, for example, would influence your behavior. And for that, I will be talking about a project that I did together with Emma Stewart. And the idea that previous experience could influence your action is not a really new one. So in this nice illustration from the group from Daniel Walpert, you see an example for that. So if you imagine you're this tennis player here, you have some sensory information where the approaching ball might land, but you also have some prior assumption based on how your opponent, for example, played before, or whether it's a good opponent or a bad one. So you could assume that they will try to play the ball very close to the line. And you can combine your previous experience or your prior assumption with your sensory input and use these together to control your action. And that this actually works on a very short time scale is debated or discussed in a recent field of research, which is focused on serial dependence effects. And here, the first study, or one of the first studies that showed something like this, is the paper by Fisher and Whitney from 2014. So in this task, people basically had to judge the orientation of a stimulus, and you see the error in this trial on the y-axis. And what you see is that the error is actually not random, but it is related directly to the orientation of the stimulus that you have seen in the previous trial with an attractive serial dependence effect. So if something you have seen in the previous trial was more counterclockwise, you're also more likely to make an error in a counterclockwise direction on the subsequent trial. And it has been debated for quite some time now, what is actually the relevant signal that is mediating this influence of previous experience. And there has been a nice paper from the group from David Burr, where peripherally again, you had to judge the orientation of a stimulus. So in this case of the central gabor here. But what they did now is they added these flanker stimuli. And what that allowed them to do is change the perceived orientation of the central stimulus while keeping the physical stimulus the same. So if these flankers are aligned, basically they create some sort of perceptual illusion which shifts the perceived orientation of the central gabor. And then by dissociating the perceptual orientation from the physical orientation, they repeated a bunch of different cry conditions and basically could conclude in the end that the signal that's mediating this effect is the perceived orientation. So it comes at a rather late point in the visual processing hierarchy, where the spatial context of this flankers is already integrated. While this was going on for perceptual decisions, something very similar have been proposed for oculomotor control. So here now is a study from the group from Steven Lisberger, where you always had two subsequent movements. The first one, the prior trial or n-1, could move fast or slow, and the following movement, the probe, always moved at the same velocity. So what you can see now in the plots here is the eye velocity in the probe stimulus. So there always the same physical stimulus was presented. However, if you have seen something fast before, your eyes also move faster in the next trial. And if you have seen something slow before, your eyes move slower in the next trial. And what they also found that the size of this effect is actually manipulated or mediated by the reliability of the signal. So if the probe trial was a low contrast stimulus, which is now the low reliability condition, you relied more on your previous experience than if the probe stimulus had a high contrast. That suggests that there is a seemingly sophisticated weighted integration of past experience and current sensory input. And what we were wondering now is, okay, so what is the relevant signal for oculomotor control again? Because again, it could be perceived speed, or it could be the retinal speed, or something else. So in these classical paradigms with moving dots, usually what you have is that retinal speed and perceived speed are always correlated. So we thought about whether we can create a situation where these two actually can be dissociated. And in the real world, they actually often are. And for that, we turned to the famous Ponzo illusion. So here, because of the perspective depth cues that you have, although these two cars actually have the same retinal size, perceptually, they look different. So the car in the background hopefully looks slightly bigger to you than the car in the foreground. And also the velocity of these are perceived differently. So we leveraged that illusion and created actually a very similar paradigm to what I've just presented to you. So we again had a prior trial and a probe trial, so two subsequent movements. The prior could be a car that was either moving fast or slow, or importantly, either in the perceived foreground or in the perceived background, despite always having the same retinal size. And that then was followed by a probe stimulus, which could either be a car now in front of a gray background, so without any additional cues, or as a control condition, a completely unrelated Gaussian blob. So to give you a feel for these data, I will now show you the average eye velocity now in the prior and in the probe trial. So for the prior, you can see that people were able to track these cars, right? So in the reddish colors, you see the fast prior, and there the eye velocity was much higher. In the bluish colors, you see the slow prior. So they were just tracking basically the movement on the screen. For the probe stimulus now, you can notice that we can replicate the previous effect. So your eyes again move faster if you have seen something fast before, and they move slower if you have seen something slow before. And we can quantify this effect by just taking the difference between the fast and the slow prior. And these values are positive, which means, again, this attractive serial dependence effect. But as you probably already have guessed from the plots, there's not really a difference between whether the car was perceived in the foreground or in the background, despite that having an effect on your perception. And one interesting side note here is that for the probe stimulus, we did not only have that car, but we also had this unrelated blob. And if you think about what these previous experience might tell you is that you could, for example, use it to learn how one car moves to predict how another car moves, but you not necessarily should use it if suddenly now a different stimulus comes along. But what we found is actually the same effect, and it was even slightly stronger. So people basically integrated the previous trial where they saw a car to update the oculomotor behavior in the following trial where they had an unrelated Gaussian blob stimulus, and the effect is even slightly stronger, probably because the blob was much lower contrast than the car, again suggesting this weighted integration of information. So yes.
Alexander, can I ask a question?
Sure.
I see that the slow condition is shifted for, in the probe. Both the slow and the fast are very close, or seem to be at least closer to they're not somewhere in the middle, but rather are shifted towards the fast speed. So while in the prior, there's a big difference between, maybe I'm misunderstanding this, it seems to me that in the probe, there is a significant difference, I understand, but it's much closer.
Yes.
The slow is closer to the fast.
There's one important detail which I haven't pointed out correctly. In the prior, there were actually two different physical target speeds. So the prior could actually move either at 5 or at 15 degrees per second, which is the red and the bluish colors. in the probe, always the same velocity was presented. So all of these targets actually moved at 10 degrees per second. And the only difference now is that before they saw either a fast or a slow trial. That's why they're much closer together. So they were tracking the same stimulus, just what they saw before differed. Yes, yeah.
Okay,
I hope that is clearer now.
Yeah, thank you.
Okay, good. So here, basically, this gives us some indirect evidence that the change in perceived speed of these cars might not really play a role. But we can actually directly test this. And for that, we had 3 different conditions now. So we had the car in the foreground as a comparison. And then we had one condition which was retinally matched. So the same car just shifted in the perceived background. And one car that was perceptually matched. So before the experiment, people actually could adjust the size and the velocity of this car so that perceptually it looks to them exactly as like the foreground comparison. And then we can again look at how these different priors influence your probe eye velocity. And the way I will be showing you this is we will look at the difference between the retinal match and the comparison and the perceptual match and the comparison. And what you can see is that the retinal match and the comparison are very similar, reflected here by a difference close to 0, whereas the perceptual match always produces a slower eye velocity on the following trial. And this behavior can actually be nicely predicted by a model that only takes into account the retinal velocity on the previous trial, suggesting that the mediating signal here for the oculomotor system really seems to be a low-level retinal velocity error from the previous trial. So to summarize these results, across this field of zero dependence, people have shown it across basically almost all levels of visual processing. So for example, from faces to very low-level oriented gabors. However, there seems to be kind of a dissociation, or at least in the comparison that we can make here, between how you use this information, for example, for perceptual decisions and oculomotor control. While this previous work has shown that basically for perceptual decisions, you integrate spatial context, so it's based on the perceived orientation in that case. For oculomotor control, we see that the prior rather seems to originate at a very low level, so low-level velocity error that is then mediating the subsequent effect. And we would argue then, rather just reflecting a dissociation between these two, this actually nicely matches the goals of the two systems. So if you think about it, for your perceptual system, it is very important that you constantly integrate and interpret what's on your retina. So only in that way you can achieve, for example, size constancy, so that if something moves in depth, it gets smaller on your retina, but you still want to perceive it at the same size. We have to achieve something as like color constancy, so we have to discount changes in lighting. So it is very important that we constantly interpret our perceptual world. Whereas for the oculomotor system, the main goal is to bring and keep objects of interest on the fovea. And for that, the critical signal is basically the retinal error. So how far do I have to move my eyes that I can bring the object of interest onto the fovea? So therefore, here the oculomotor system seems to use the relevant information for it, which is in this case the retinal information, to achieve a tracking goal. So if we now go back again to our initial list of potential differences between these artificial experiments and more naturalistic behavior, we can see that, again, previous experience as we have with the world, for example, in terms of size constancy, but also even on a short time scale from these trial-by-trial things, we do not typically capture if we, for example, look just at average behavior in condition 1 versus condition 2. And when we're looking into that in more detail, we can find some interesting things like this dissociation between action and perception. Okay. And that now brings me to my last part that I want to tell you about, which is now focusing on more complex naturalistic stimuli. And this is work that I have done again together with Karl Gegenfurtner and Doris Braun in Gießen, and a group from James Elder in York, mainly together with his PhD student Hemant Pitapati. And for that, let's think back again about this step ramp paradigm. Here, a target moves across the screen, And usually what it does, it moves even at a constant velocity. So you could argue again already, so, well, in the real world, there's never something that actually moves at such a constant velocity for a constant time. So why wouldn't it be more useful to study complex movements, for example, like this moving dot here? And you might already notice that tracking such a moving dot is actually already much more difficult than, for example, following a continuous motion across the screen. But you could also ask, well, whenever does something like this actually also happen in the real world? Maybe you have experience with that, like when you try to catch a mosquito in the evening, but when does it happen besides from that? And the answer is that actually doesn't happen that often. We just don't really have a good impression how things would look in an isolated sense on our retina. So this now is actually the movement of a puck in the ice hockey game, which I hope you can see over Zoom and with all of the potential lag that might add, but it's actually the same low-level motion trajectory. So we had these ice hockey videos from the group from James Elder with hand-labeled puck positions. And that allowed us to basically isolate the movement and then compare how is your tracking performance when you just see the motion trajectory in contrast to when you see the motion trajectory and it's basically in its natural context. So for that, oh, sorry, that was one too far. We created 3 different conditions. One where you just see the disk, as I just have shown you, or one where you just see the original video, even without this little circle that I added on top of it for you to make it easier to find the puck. And we had one intermediate condition where we just showed a static frame and then played the puck on top of it. And 15 non-expert observers watched these videos. And it is important to note that all of these scenes were actually filmed with a static camera. And the reason why I'm pointing this out is that in your typical sports broadcast, what actually will happen is that the camera pans with the action. So most of the action actually will take place in the center. So you don't really have to move your eyes, but basically the camera is doing the work for you. Whereas in our case, people actually had to move their eyes around. So now we can compare tracking behavior across these 3 different conditions. So initially, we just looked at, for example, standard metrics such as the position error. So how far is your eye away from the puck? And across all of these, across all of the conditions, people were quite good in it. And there was a slight increase in the position error for the complex contextual video. And that might not really come as a surprise, but what we did next was we tried to determine the latency in these complex videos. So how fast are you reacting to a change in the trajectory of the puck? And we did this via a technique that is called cross-correlation. And for those of you who are not familiar with this, what you basically are doing is you're taking the vector of the target and the vector of the eye, and you're shifting them relative to each other. And the point in time where they have the highest correlation is basically your estimate of the latency of the 2 signals. And for the disk condition, you can see that with that, you estimate a lag behind the target trajectory by about 180 milliseconds, which is a typical eye movement latency. So if I would show you just a dot flashing in the periphery and you would need to make an eye movement there, it probably also would take you around 200 milliseconds. However, when we repeated this analysis across the other conditions, we found a very striking difference, which was that in the video condition, the peak here was actually shifted to 0. So on average, you basically had no delay between when your eyes reacted to a change in the target trajectory. And sometimes you were even in front of that. And while this is a bit hard to imagine when you just look at a cross-correlation, let me give you an example. So we specifically looked at passes between players. And here you see a still frame where the player in the black jersey in the bottom right of the image is passing the puck to the player in the center of the field. And I have added some example trials for the different conditions as these squares. And one thing you might already notice is that all of the purplish squares, which reflect the video condition, are by much further ahead than all of the other squares. And some of these people are actually even in front of the puck, which is this black circle. And that this is not just a coincidence, we can actually quantify. So what we did is we measured the time when people arrived at the player that is receiving the pass. And you can see again that there's a drastic difference between the video and the two other conditions. So people arrive there roughly 150 milliseconds earlier if they see the video, and they arrive there even before the puck on average, which would be the dashed line at 0. And interestingly, they do not only arrive there earlier, but they also arrive there more accurately. So while in the video condition, they basically fixate directly on the player who's receiving the puck. In the other two conditions, they basically always overshoot the player because they don't know when the pass is stopping. So they extrapolate, and once they notice it, they're already basically over the location where the pass was stopping. So to summarize these results, that understanding a scene has a lot of influence on how you move your eyes, because it allows you to make concrete predictions where you should look. And this increased error that I have shown to you in the beginning, so the increase in position error, actually is also based on the fact that they sometimes just do this prediction, because if you look ahead, that will produce actually a large position error. But it is also a very smart strategy because it allows you to be with your eyes at the right point at the right time. So where the decision is happening. So what's happening next is basically determined by the player who's receiving the pass. So if you can fixate there already to basically see what's going to happen next, that is a really useful strategy. And this is also reflected in some other work that has shown that you can basically integrate your knowledge about the world into how you move your eyes. So for example, if you have statistical structure, Eileen Kauler has shown that there, for example, is anticipatory eye movement behavior, or if you have to track a moving ball, if it behaves according to gravity, you are much better in tracking it than if, for example, it would obey some other strange rule of even inverse gravity or a different gravity constant. So that suggests that basically we can use our contextual information to counter to typical internal processing delays that we would have if we would simply need to react to something. And that now brings me to the last two small pieces of work that I want to show you, where we looked at what actually drives this predictive advantage. So in this study now, we had the two extreme cases. We had the disk in front of a gray background with no contextual cues, and we had the video with every contextual cue imaginable available. So what we tried now is to piece apart basically which of these types of information actually might be relevant. And this is work that was mainly driven by two very excellent students, Linus Leske and Niels Borgeding. And on the one hand, we looked at what are potential cues that could be useful in achieving this predictive tracking effect. And for that, for example, we just showed you the positions of the players without giving you any access about the kinematics by simply replacing the players with squares. Or we manipulated the kinematic cues by, for example, playing the whole video in reverse, which really impaired the causal structure of the kinematic cues that were available. And to show you a brief glimpse into these results, we had a total of 6 different conditions. On the two extremes, there was the disk and the video that you already have seen. And then in red, there were the 2 contextual conditions where we just replaced the position by players with squares. In the one condition, we even showed you the teams, and the other condition, we didn't do that. And then in the 2 bluish conditions, we manipulated the kinematic cues. So we either played in the video in reverse, as I've just shown you, or we flipped the video vertically. So everything was upside down. So you could still be able to analyze how people are moving, but it was just a bit more difficult. And the results, in short, are that only knowing where players are, so the contextual cue conditions, they actually did not lead to this predictive eye movement behavior that we saw in the original videos. Playing the video in reverse really messed with people. So they got even worse than in the disk condition, because basically by playing it in reverse, you completely impaired the causal structure. So someone who's playing a pass now suddenly looks like he's receiving it and then suddenly does something like this with the stick. So they basically constantly were making wrong predictions, and that led to even a stronger lag behind the disk. And when flipping the video vertically, where it was just a bit more difficult to analyze the player's movement, people were actually surprisingly good. So they were very close in the performance to seeing the original video, which suggests that as long as you somehow can analyze how the players are moving, you can use this as the main cue to predict what's going to happen next. In the second half that I quickly want to mention, we looked at expertise effects and how much information you basically need to achieve this predictive tracking. And for that, we replayed the video with now different cutouts around the puck, so to give you access to different amounts of available information. And we again had 4 conditions now, so the disk and the video as our controls, and then the 2 intermediate solutions in the middle. And we recruited 2 different groups, one again a novice group, and another group of expert ice hockey fans who basically said they were constantly watching, they might be even like part of a fan club of a local ice hockey club and so on. And then we compared basically the tracking performance of those as well. And what we found was that overall, the more peripheral information became available, you had a stronger predictive tracking effect. So, and it's interesting to note here that even the video showed a better predictive tracking than the large cutout, which was like a Gaussian window of about 8 to 10 degrees. So even information further out in the periphery than 10 degrees still helped you somehow to have a better predictive tracking behavior. And in general, we found that experts overall showed better tracking behavior than our novices, but there was also an interaction where them especially benefited even in these intermediate conditions from the additional cues. Okay, so to conclude across all of the studies is that we have seen in the first study that behavior can be tailored according to your own strengths, and that our previous experience, how we use that actually also reflects the goal of the different systems that we are using. And we have seen that context can play a very critical role for ocular motor control. So we basically started with this question. So how well can we generalize our typical results in these artificial experiments to how we behave in a natural world? And what especially the last example is showing is that ocular motor control in the real world is more than just following dots. It's more than just reacting to stimuli, because it's highly influenced by our understanding of the scene. And this understanding can be seen in, for example, that you coordinate your movements according to your own strength. So you basically know what's going to happen. You integrate your previous experience into your behavior, and you can use these contextual cues to guide your eyes to the right place at the right time. And with that, I want to thank you for your attention. I want to quickly acknowledge all of the people who did most of the work with me and our funding agencies. And with that, I'm very happy to take questions.
Thank you very much, Alexander. It was a really interesting talk. So I'd like to ask everybody to unmute yourself, and let's give Alexander a big applause for this very exciting talk. Well, I'm hearing only myself. Yeah, thank you for joining me. Maybe some people haven't unmuted. So definitely, I'm sure people will have questions. I'm going to start myself before others have a go. So I wanted to ask with respect to the peripheral information, or the fact that you see that the more peripheral you have, these are exciting results, but I want to ask if you think, I mean, because the players themselves have also some motion that we are used to seeing. I mean, it's not just the overall motion, but within themselves, you know, they use the hitting. And we somehow, so I wanted to ask if you carried out an analysis where you looked at when there was proper or correct passes versus incorrect passes, because maybe we assume that there's going to be a correct pass, so we already jumped to the other player where the pass is headed to. But maybe if, you know, sometimes passes are incorrect, and maybe then there will be more errors. I'm just wondering if we use that information to guide us to, and whether the eye movements are, you know, similar in their velocity to, let's say, the target's own velocity. So do we make saccades in the, you know, speed of the actual target?
Yes. Okay. I think there were a bunch of questions, but let me try to answer them. So what we basically see is that we don't, unfortunately, do not have enough like fake passes in the videos that we had to like separate the analysis based on this. But what we did find is when in the conditions where you had different amounts of available peripheral information, when we basically align the passes with how well the target, for example, was visible, so the passes would have different distances, you can clearly see a trend so that only if the peripheral, the pass target basically was available in the periphery, then you saw this immediate predictive advantage. So then basically what people are doing is as soon as they see that someone is initiating a pass, they try to estimate where it will be going. So if they see a player somewhere in the periphery, they will quickly follow the puck and then make a saccade to look at the player who's receiving the puck, and then just stay there to wait till this person receives it.
I see.
Another thing that we noticed is that actually, if you think about the disc and the video, there was also large differences in how people move their eyes. So in the video condition, you would see much fewer saccades than in the disc condition, and you see more pursuit in the videos. And we initially thought in the first study, we thought that this would be related again to like prediction. So you need like a lot of small corrective saccades in the disc condition to keep track of things that you just don't need if you are able to predict what's going to happen next. But what we actually found that this was also the case, for example, for the reverse condition, where people were actually much worse, where they didn't show that predictive advantage. So what we think is driving like this difference in behavior is actually something that for the disc condition, you have a very clearly defined positional target. So there's like one single dot on the screen that you have to track. So it's very easy to basically have some kind of a position error signal. Whereas for the video, there's much more going on and it's much more motion and like the players are larger. So you might not need to correct as precisely as for the disc condition. And that drives the differences in eye movement behavior.
Thank you. Very interesting. Maria, do you want to ask a question?
Yes. Hello. Hi. Very interesting. Thank you very much. I'm not in this field, but I found it very fascinating. So I have a very maybe naive question. I was wondering if there are any study for kids that have nystagmus, how they would, I'm speaking about moderate nystagmus because, of course, if you have a serious problem, you don't have very vision or you have to undergo surgery. But I wonder how, the development of these kids is influenced through this, eye movement that do not work properly. And therefore, everything you've said might be completely changed in these kids. Do you have any feedback on this?
Yes, I must admit that I do not know that much about nystagmus, so I'm afraid I can't really answer that. I don't know if any study that would link, for example, like how you perceive a stimulus with nystagmus. What we are currently trying is to, for example, relate these findings not necessarily to like nystagmus, but for example, other psychological diseases. So it is, for example, known that in schizophrenia, for example, in particular, pursued eye movements are impaired. And these are typically studied again with these very simple moving targets that are moving back and forth. So there, like a reduced pursuit gain, for example, has been floated as a potential biomarker that you, for example, would also find in relatives of the people who actually get the diagnosis. And we were wondering whether these might actually be even stronger impaired if in these contextual cues, or whether, for example, other diseases who might not rely that much on prediction. So, for example, autistic patients, for these ideas have been floated, that, for example, in the reverse condition, they actually might not be as strongly as impaired as normal people or like the regular population. So I think in general, there's a large debate on like saccadic and pursuit eye movements in general, tracking behavior, how these could be linked to potential biomarkers. For nystagmus, I'm afraid I don't know too much, so I can't really answer.
Thank you.
I think you're still muted, Sharon.
Yes, thank you. I was. So first of all, I asked if there were more questions, and then I saw that Suresh Krishna wrote, please ask for me, so I will. If there is time, yeah, well, there is time, right? Okay.
Yes.
Can we equally well say that the strength of the observers emerge from the behavior of the observer via lifelong practice, or have causality be in both directions? I don't know if that was clear or not.
Yes, yeah. So I think it is probably referring to the first study where we saw that there's this correlation between saccadic and pursuit performance. And it's a completely fair point. With the results we're currently having, we actually can't say, it's only a correlation. So we can't really say anything about the directional effect. So it could be that either basically you initially just by accident used more pursuit, and therefore you are more trained to do that. So your pursuit over time got better, and therefore it's just established at that. Or it could be the other way around, that by default you had like different accuracies in these two types of eye movements, and over time you learned which of them is better, and therefore you use them more. So that we can't unfortunately really say with our correlational data. I could imagine both. I think it's just fascinating that basically these individual differences that are often treated as some kind of noise if you compare across different conditions, for example, that these actually are very meaningful. So, for example, there's a study with a similar idea from the group from Miguel Eckstein, where they looked at where people fixate on faces. And they actually have found as well that where people are looking can differ. So some people look closer to the eyes, and some people might are looking closer to the mouth. But what they have shown is that basically where these people are looking, it's kind of their individual optimum to discriminate faces. So the person who's looking closer to the eyes is also better in the discriminating faces when they are looking closer to the eyes, instead of to like a theoretical optimum or to the location where someone else is focusing on. So I think these individual differences here are, yeah, a very useful source of information that can be very interesting to study across like different tasks, or across, yeah, it makes a lot of sense to understand where they're coming from, I think.
Okay, wonderful. Now I've got another question, which probably, or maybe the last. So you're talking about past experience and context, and I actually think they are more or less very, if not the same, because, you know, context is something that we learn through our lifetime experience. You know, I assume, maybe I'm wrong, that, you know, very young children maybe have much less contextual influences because their past visual experience is different. Anyway, what I wanted to ask was whether perhaps you thought, or maybe you already are, investigating these studies, and especially maybe in the video, with children in various ages, because, you know, if you want to see, I mean, I'm not sure it will, you know, directly show you some causal relationship, but clearly maybe this is something that is developmental, and you can see an age effect of how, you know, the videos affect, and maybe you will see that in children, I don't know, the crop outs of maybe the videos will be the same, so maybe our peripheral vision changes, or maybe we'll see some differences in the different ages that may be interesting, although it's not directly causal, but still it may give some indication as to what is contributing. So just idea.
Yes. Yeah, I think that is a very good idea. I mean, we haven't started it with like from a developmental perspective, but what was really surprising to us that is like that even novices who have no experience with ice hockey, like they show almost the same effect. I mean, there's some expertise effect on top of that, but most of the effect seems to be driven by some kind of intuitive understanding of how the world works, or like some intuitive physics that if I hit something with a stick, it will go in that direction, or how people are moving. So I can clearly see that there could be a relationship between basically when these abilities develop and how people would track, for example, puck in an ice hockey game. So I think it's a really interesting point because, yeah, most of the effect seems to be driven by some kind of like intuitive representation of the world. So I think that would be really nice to study.
Cool. I mean, yeah. Okay, wonderful. And if there is time, there's an additional question in the chat by Veronica Lukianova. Yes.
Mm-hmm.
Okay, so she says, could it be that those individual differences in smooth pursuit and saccades are related to the cone density, meaning retinal sampling capacity?
Yes, clearly. So, I mean, I think, again, so we only observe these differences on a behavioral level. Where the differences in saccadic and pursuit accuracy actually come from, we are not really sure. And I mean, one of the reasons could definitely be something that the sensory representation of the stimulus might differ. It could be that at some point in the processing stream, there might be different noise sources added to either one or the other. It could mean that they actually are somewhat like separate systems, but we have enough evidence that I think that is rather an unlikely situation. Yeah, overall, where these are coming from is kind of up to debate now. And I mean, I think there, like this individual differences approach again might be like something very reasonable to do, because if you have access to, for example, cone density measurements for these people, you could see and just try again with like a correlational approach, see whether these were, for example, the people who are more accurate in making saccades, or these are the people who are making more saccades. So I think there's a lot of potential room to basically grow these ideas of like how individual differences across tasks are unfolding and what we can learn from those.
Definitely. So Alexander, thank you so much for very interesting and insightful ideas and directions and novel
thank you
research.
Very much for having me.
Yeah, it was a real pleasure. And I want to say that next week, we will have Domenica Bueti, Professor Domenica Bueti, who will be talking about a sense without sensors, how non-temporal stimulus features influence the perception and the neural representation of time. So you're welcome to join us. And again, thank you very much for the very interesting talk. And see you at VSS.
Yes, thank you. Bye.
Thank you. Bye.